الرحمن الرحيم. في البداية بحب أشكر شركة فاكسين فالي على إتاحة الفرصة إن أنا أبقى متواجد معاكم. الحقيقة النهاردة أعرفكم بنفسي الأول. أنا دكتور كريم عيد، مدرس أمراض الدواجن، كلية الطب البيطري جامعة سويف. حصل على درجة الدكتوراه من جامعة برلين بالتعاون مع المعهد الفيدرالي الألماني لصحة الحيوان. الحقيقة كان عندي فرصة كويسة جدا إن أنا أشتغل في المعمل المرجعي للانفلونزا في ألمانيا لمدة عامين، فده أتاح لي فرصة كويسة جدا إن أنا أبقى متواجد وإن أنا يبقى عندي اكسبيرينس كويسة في الانفلونزا. والمرة الثانية حابب أشكر دكتور محمد عادل ودكتور عبد الرحمن شاهين وكل شركة فاكسين فالي على التواجد النهاردة. الحقيقة عنوان المحاضرة النهاردة Low Pathogenic Avian Influenza H9N2 A Continuous Challenge and Molecular Evolution. العنوان ده احنا قصدينه او انا قصده، ومن خلال المحاضرة هنشوف ازاي ان اللو باثوجينيك ايفيان انفلونزا هو عبارة عن continuous challenge وايه الموليكولار ايفوليوشن اللي حصل فيه في خلال السنين الاخيرة. لكن عشان نقدر نتكلم عن الكلام ده او نشوف الكلام ده، هنشوف ان البولتري برودكشن في الميدل ايست في وافريقيا عموما في تطور مستمر. ان حوالي افريقيا والميدل ايست بينتجوا حوالي 17.8% من البولتري برودكشن around the world. I can switch to English if you have other colleagues from other countries to can understand us. Poultry production in Middle East and North Africa. We can see here in this chart the total human population of Middle East and North Africa in 2016 representing 7.4% of the world population. And consequently, the region also produced 2.4 billion pairs in 2014, about 10.4% of the total poultry production worldwide. الحقيقة ده معناه إن إحنا عدد السكان في تزايد مستمر، وإحنا محتاجين كمية كبيرة جدا من البولتري، وهو ده considered the cheapest source of animal protein is poultry, and still we have a gap between the human demand for animal protein and poultry production. And here we can see that most of countries which we are talking about, the Middle East, North Africa, and the amount of the poultry production and the population number and size of each country. In Egypt, we have about 102 million of human population, and we have investment about 3.7 billion dollar, and also we have 2.5 million of workers, and I think we have more than 2 million of other workers with indirect deal with poultry production in Egypt. So we have huge investment, a lot of people working in this industry, and also we have a lot of humans that need cheap source of animal protein. And also here we can say that the poultry production in Egypt is huge industry. We can see in this slide a lot of grandparents, breeders, one-day-old chicks, broilers, ducks, and a lot of other species. So the poultry production in Egypt is in continuous elevation, and it was really good industry and investment until the avian influenza introduced and appeared in Egypt in 2006. Actually, Egypt and Middle East, and I think most of countries suffering from different respiratory viruses like avian influenza, Newcastle disease, infectious bronchitis virus, and other viruses. But the most important one is avian influenza virus due to its zoonotic nature, especially the highly pathogenic one. And the avian influenza genome, we all know about avian influenza. It is genomic structure is a little bit different. It has 8 separated segments of nucleic acid, and this allows the avian influenza to reassort and the continuous evolution. And the most important two proteins in avian influenza that considered control the subtype of avian influenza is H9N2. We have 16 HA and 9 NA subtypes from avian influenza. And here we can say if two viruses of avian influenza insert to one cell, we can have 73 possibilities of new viruses that can evolve from the reassortment of those two viruses. In Egypt, avian influenza history and the story started from 2006 when the H5N1 221 first detected, and then here we can see that using of vaccines that have mismatching with the field viruses leads to evolution and of the skip mutant viruses and also nucleates 2211. 2014 2212 of H5N1 evolved also, and it was really dangerous because it had high possibility to transfer to the human. And for the low pathogenic avian influenza, it's first detected in Egypt in 2011, and who circulated with H5N1 for many years. At 2016, H5N8 first detected in Egypt, and I think it was the global outbreak of H5N8. And also at 2019 and 2020, the H5N2 first detected in Egypt. Actually, the situation is really complicated due to we have endemic infection of avian influenza, we have lack of biosecurity measures, we have a lot of live birth markets and transportation of live births between governors. And Middle East, north of Africa, and Gulf area is in the main ways of migratory birds, and it is really, and it was really dangerous because the reassortment possibilities, and it's also it's really it's really the main reason for continuous introduction of different viruses into these countries. And also the migratory birds with presence of backyard, it is very easy to exchange and transfer of new viruses to this backyard, and this backyard with its contact with domestic birds, so it is the way of introduction of new viruses from migratory birds, backyards, then domestic. In Egypt also we can see in this slide that we have the most of people are living on the both sides of Nile River, and also the poultry farms are in the same place. So it's very dangerous due to we can't control the avian influenza infection and also the transfer of the virus to human population. The main proteins responsible for avian influenza replications, HA, matrix, and those are the surface protein, and also the NP protein is playing a role in the virus replication. And the main difference between the highly pathogenic and low pathogenic avian influenza is the cleavage site. We can see that the HA is the main determinant of the low or high pathogenic avian influenza by presence of this cleavage site. If this cleavage site is really long, have multiple basic amino acids, so any furin proteins or proteases can cleave this place and start the replication cycle of avian influenza. But if this cleavage site is really short and have 1 or 2 or 3 only amino acids in this place, so it is very hard to cleave it. So need a special type of enzyme, which is trypsin-like enzyme, which is only we can find this in the respiratory and intestinal tract or cells of the poultry. So it's the place of replication is paired only in the respiratory and intestinal tract. When the enzymes is cleaved, the HA, so it is the HA2 work as the injector and transfer the NA nucleic acid inside the cell and start the cycle of the replication. And this slide also that we can say that not only the host cell proteases is important, but also some others proteases that we call exogenous proteases. Exogenous proteases, it can be from the bacterial origin or others like IBV, coronavirus that have trypsin-like enzyme in their envelope also play an important role in increasing the replication of the virus. So, low pathogenic influenza history actually started in USA in 1966, and based on the HA sequence, the H9N2 viruses, we can say that we have or grouped into several genotypes, about 4 genotypes, G1 like that we have in North Africa and Middle East and Gulf area, and Y 280 like Pyang 94 like and Korean like. We can found here that all this blue area are G1 like, and that we have in Gulf and North of Africa and Middle East area, and these others for other colors for other genotypes. Also, we can see that this Russia that have G1 with Pyang and together, and also this in China G1 and also Pyang. The G1-like lineage is also divided into 4 types, A, P, C, D, according to the phylogenetic analysis of all genes segments. And here we can see that this P type that we have in Egypt and recent isolates from Lebanon, Israel, and Qatar, and UAE, Afghanistan, Iran, Tunisia. This is all from P, and others played a little bit more old in Israel, UAE, and Iran, and Saudi Arabia, Emirates. Actually, we can say that H9N2 is a thief. It's like a thief. It's widespread in most of poultry farms and leading to respiratory disease, drop of egg production, co-infection, immune suppression, and reassortment also as a general donor. Actually, in Egypt, the H9N2 is widely distributed. In this study at 2015 and 16, about 92% of the surveilled or from the farms that use in this surveillance were infected with H9N2, and also in other studies that we did in poultry farms in Egypt at 2016 and 2019, we can see here in this study in 2019 that these yellow colors in different Egyptian governorates that have a high percentage of detection of H9N2. And also in this study, we can see that 27 farms are positive for H9N2 from 39 farms. Actually, this percentage near 70% of the farms were infected with H9N2. H9N2 is not have a pathognomic clinical signs, normal respiratory signs, coughing, sneezing, lacrimation, face edema, depression, decrease in the water and food consumption, and the mortality rate is low if it's single infection, but with the co-infection like other viruses or bacteria, we have high mortality rates actually. Signs and lesions in the co-infection are more clear because no pathognomic sign, no pathognomic post-mortem lesions in the single infection. But in the co-infection with especially with IP, we can find cyanosis in head, some mucus and tracheitis. We can also find enteritis and some redness in shank, nephritis, and caseous plaque. Actually, H9N2 in the single infection, and it is proved in also a study that we did in 2016 and 17 that we can find that the H9N2 single infection can leads to partial caseous plaque in tracheal bifurcation. Also in the preterus and layers, the H9N2 infection in ovary and oviduct can produce dropful or cause dropful egg production, and this drop of the egg production reaches to 30 to 40% within 2 weeks and can be recovered from 3 to 5 weeks, but the curve of the egg production not came back to its normal. Again, the quality not affected, but maybe the reduce in the feed intake leading to decrease the size of the egg. Lesions is also not pathognomic in the previous layers. We can find ruptured ova, free yolk, inflammation in the over and oviduct. We can find in some cases nephritis. The co-infection is really important in the H9N2. The co-infection either with bacterial, Mycoplasma, E. coli, Aphibacterium, ORT, or viral like Newcastle and IBV, leading to change in the behavior of the mortality rate in the flock and leading to increase the mortality rate. Due to these bacterial diseases can produce proteases, trypsin-like enzymes that we already said, so it is started the replication and increasing the replication of the virus, and maybe also in other places, if this bacteria found in the liver and the kidney, they can produce a trypsin-like enzyme, and we found that the H9N2 can replicate in not target organs that the virus is normally going to due to presence of trypsin-like enzymes that produced by external exogenous proteases that we can say. And also the IBV is really important. IBV, even the classical or variant strains, or even the vaccinal strains, due to the IBV is a coronavirus, and the coronavirus have in its envelope normally trypsin-like enzyme. So when we found the IBV as a virus, even vaccinal one, it can exaggerate the low pathogenic avian influenza H9N2 cleavage and replication. So we can see here in this slide the synergism between the low pathogenic avian influenza and IBV. IBV, infectious bronchitis virus, leading to normally even the vaccinal one, especially the variants, the cellulosis and deciliation, and providing trypsin-like enzyme or proteases that included in the coronavirus envelope, leading to increase the cleavability of the H9N2, leading to thymus and spleen atrophy and immune suppression that came back again to the IBV to increase its replication. And a lot of studies actually focused on the role of some bacterial infection like Mycoplasma, like ORT that we already said, and Aphibacterium and E. coli in the exaggeration, the H9N2 replication and mortality. And also we have this study in 2017 about the role of the co-infection between the H9N2 and IBV, even the vaccinal one, and we found in the single infection the shedding titers in the single infection decreased after 7 days, while the H9 groups that have classical variant IBV and even the vaccinal one, the shedding titers is increased in comparison to the single infection H9N2. That proved the theory that the increasing the virus replication and extension the period of shedding. Actually, to understand the effect of the H9N2 and its effect on the immune system, we can follow this slide. The inhalation of the H9N2 and virus enter into the receptor binding sites in the respiratory and intestinal tract, which cleaved and increase the virus replication, then enter to the host microbiome. So all of influenza make lysis, but the H9N2, when entered the macrophage, the NS1 structured protein 1 suppress the macrophage, leading to stop the apoptosis process and uncontrol of the virus replication, and also leading to inhibition of the interleukin and CD4 and CD8 T cell, leading to cell. Actually, the T cell is, we can say, the maestro of the immune system regulation. So when inhibition of the T cell, it's also inhibition of the immune system process and inhibition of the antibody production process from the plasma cell. Here we can follow this, and non-structured protein stops the apoptosis of the macrophage and control virus replication, down regulation of interleukin 4, inhibition of the CD4 and CD8, and also inhibition of the human immunity consequently. And other studies proved that the H9N2 infection leading to lymphocyte necrosis in the persa and thymus, especially thymus. Actually, also in our study, we found this. So we can say the effect of the H9N2 will be general in the immune system. That we found that the vaccinated person with Newcastle, the level of the antibodies of against Newcastle is decreased when the person infected with H9N2 in comparison to the control group that vaccinated non-infected. That means that the H9N2 is make general immune suppression. And I think this, we also followed the histopathological score in our study, and here we found that the thymus is really affected by the infection with H9N2, and we can also found this in the these figures. This group from D to G are infected even H9 or H9 with IBV, and we found the thymus have hemorrhage and necrosis, and after this we found this thymus are already atrophied in comparison to the control groups and single infected with the IBV without H9. And this part was about the continuous challenge of H9N2, but about the genetic evolution, we usually focusing on the HA and the NA evolution of the H9N2 without focusing on the other genes. Here, even in the HA and the NA evolution, HA and NA genes, we found evolution in these genes, meaning this evolution about a lot of mutations. And also in this study that we did in 2019, we found that all viruses from 2015 until now, from the H9N2, the HA is already evoluted and formed a subclade. These subclades are characteristic by some of amino acid changes. And also in the NA, we found also this subclading, and this was especially in 2017 and 18, we have found N-glycosylation, acquired N-glycosylation site that was found in the H9N2 of human origin, that meaning that the virus is acquiring the possibilities of transfer, more ability to transfer to human, and this is really dangerous. About the reassortment in Egypt, especially, we don't found any reassortment until 2014. Even the H5N1 introduced or detected at 2006, H9N2 in 2011, so they were circulating together without any reassortment. The first reassortment evenness was detected in the H9N2 virus in 2014 in pigeon. We found that in a study at 2017. And for reassortment detection, we can't only depend on the HA and the NA sequence. We need more. And the full genome sequence is very important to put or to make the epidemiological map. The epidemiological map, not only we can say that we have the numbers of outbreaks or we have subtype H5N8, H5N1, not like this. The full genome sequencing is of utmost importance, because it allows us to track in the origin of this virus introduction, providing full data to put the control strategy. We can also study the dangers of the virus and the possibilities to transfer to the human. And this genomic surveillance is enabling the rapid investigation with the evolutionary and transmission dynamic of the avian influenza in the local, regional, or even the international scale. And the most important thing is allows us to select the most suitable strain for vaccine manufacture. So we come back now to the H9N2 reassorted virus that detected in Belgium in 2014, that we found 5 segments, new segments acquired from wild birds from different birds, H3N8, H1N1, H7N3, H2N3. And actually, we in this diagram, we did a full surveillance, and we did full genome sequence analysis for H5N1, H5N8, H9N2, the whole viruses that we found in Egypt. And actually, it was a surprise for us that we found new H9N2 novel reassortant virus that already came from the original H9N2 virus that was circulating in Egypt from 2011 and 10, and we found the reassortment occurred with the new virus that detected in Belgium, and now the circulating virus in Egypt from 2016 or 17 is new virus that only have 3 gene segments, HA, NA, and M from the old virus, and other internal genes are completely new, acquired from wild birds, H1N1, H7N7, and other viruses. And actually, this may be reflected on the virus fitness, replication, and spread. And it's only the dangerous is not only in this, but the dangerous is that the new virus, its ability to reassortment. The old virus was very long time without any reassortment events, and with the new introduction of the H5N8 into Egypt at 2016, and it becomes the more predominant, and H5N1 nearly disappeared. This new virus H5N8, it's all we know about that. It's have also high ability to transfer and reassortment. So now we have 2 viruses have the ability, high ability to and possibility to reassortment H5N8 and H9N2, and that what occurred already in 2019 and 20 in Egypt, that we have new reassorted H5N2 viruses came from those 2 parents, H9N2, new reassortment H9N2 and H5N8, both occurred reassortment. Then we have this new virus H5N2. So H9N2 is a general donor. It's really important to know that the H9N2 reassortment is really dangerous, because we all know about this. The H9N2 is a general donor. And in China, in H7N9, the G1 like lead in China, the H9N2 gave 5 genes, or donated 5 genes to the H7N9, and it is really zoonotic. Okay. By the genotyping analysis of all Egyptian H9N2 full genome sequences that we have, actually we found that the H9N2 in Egypt, we have 3 genotypes. We did what we know, or what building of the full genome, like concatenated full genome sequence building, and we found that we have genotype 1, that the old virus circulating in chickens, then the genotype 2 that detect in pigeon, then the genotype 3, it was the resulted from the reassortment between G1 and G2. And here we can see the internal genes and all genome, and we found that the new genotype 3 is a reassortment between the genotype 1 and 2. And here we describe that the H5N2 reassorted virus parents was the new reassorted H9N2 and the H5N8. And actually, here we proved that the new H9N2 have a ability reassortment, more ability to reassortment, and the H5N8, it's already we all know about the possibility of reassortment. Not only in the reassortment, but also we have diagnostic challenge. The H9N2 circulations and a lot of mutations, especially in the HA, leads to failure in the diagnostic, even with in real-time PCR, and this what we discovered also in Germany in the reference laboratory, and we actually modified and improved the primers and the probe, and also published in 2019 the new primers and the probe for H9N2 detection. So we have a lot of problems, continuous risk, and also evolution, genetic evolution. So how we can deal this? The most important possible solutions that we are talking about more and more, supply security. Supply security is the most important step to control any virus introduction to your farm, including vehicles and the workers, veterinarians, car, that car feeds, water supplies, all of these things we must control and prevent. The second thing is transfer the poultry farms to area away from the human population, or away from the people who are living, to control also the virus introduction, to control the vehicle movement, and you can put good biosecurity measures in these new farms. And the last thing is the vaccination. The vaccination is very important in the endemic countries due to its control, or increase the resistance to the field challenge, reduce the shedding level also, and reduce the, if the shedding is reduced, also the transmission of the virus will be reduced, and this must be with the virus surveillance, especially the full genome sequence. And in the vaccination, the influenza, in general, live virus vaccines is forbidden due to the ability of the virus for reassortment. The maternal immunity is very important to control or to prevent the early infection in young chicks, and also the flock must be the young chicks healthy to respond to the vaccination process. Even influenza vaccines, we have hatchery vaccines and inactivated vaccines. Here in H9N2, the old vaccines, I think, inactivated. This chart or diagram is very important. It is, we can see here the relation between the maternal antibodies and the time of the vaccination. The maternal antibodies is very high during the first 2 weeks, and they start to decrease until the 21 days. I think it's more antibodies found for protection for influenza. And when you introduce your vaccine at 3 to 5 days, you can also after 21 days and about 26 days, you will have a good protective titers from the antibody, so it can protect your flock. And the period between 18 to 24 days, it is the period which we call negative phase, where the maternal antibody level is decreased and still the vaccinal antibody in its way to increased. So this negative phase is very dangerous, and it is the time that the flock can easily catch the infection. So if you increase the maternal antibody can be increased more, and also can decrease this negative phase, or decrease the time period of this negative phase, and also to avoid this side effect. So that you give only three times H9N2 vaccines to the preterm flock, you can increase it to 4 times, and this will reflect on the maternal antibody. And what about the early infection? A lot of flocks have early infection of H9N2. That's reflected or owing to some causes. Maybe the maternal antibody itself is really low. So just give 1 or 2 times of H9N2 vaccines to the breeder flock, so the newly hatched chick have low maternal antibody. Or the breeder flocks already challenged with many times H9N2, so the level of the H9N2 antibodies in the breeder flock also is low, so will be low in the newly hatched chick. Or you give already mismatched vaccine with circulating strains, so you have what we can say false level of the antibodies and in newly hatched chick, and can't protect you because there are mismatching between the strain and the antibodies that we have in your chick, so it's also easy to occur the early infection. So how to evaluate your vaccine efficacy? Maybe according to the experimental infection or field infection, you can depend on the HI test by collecting the blood sample, and also real-time PCR to detect the shedding or challenge it's already occurred or not. So the final summary of this lecture, we can say the widespread of H9N2 in poultry farms reflect the failure in the control strategy. The genotyping and reassortment analysis of the H9N2 in Egypt revealed presence of 3 different genotypes of H9N2 by the full genome sequence, and it's really important to say this. We must increase the numbers of the samples that full genome sequence analysis is used for. And the low pathogenic influenza play an important role in the evolution of the novel genotype avian influenza as a general donor. And also the heat map phylogenetic analysis found that the H9N2 have a lot of genome segments from other viruses, 77, 73, 79, strain 6 that already detected in Egypt from wild birds. The vaccination is the cornerstone in the endemic countries toward the avian influenza eradication. And I want to thank you again, and I would like and I appreciate also this good chance from the company of Vaccine Valley to be here and to present this data. Thank you.